In today's episode, exploring cutting-edge advancements in synthetic biology, we'll delve into the incredible world of biosensors, devices that are revolutionizing the way we interact with and understand our environment. Biosensors are chemical analytical devices that integrate biologically derived recognition elements with sophisticated transducers to detect and quantify specific target analytes. In short, they sense how much of something there is. Biosensors have become the cornerstone of modern healthcare, enabling rapid and accurate diagnosis of diseases, monitoring of patient conditions, and personalized treatment, detecting biomarkers, pathogens, and other important molecules with unparalleled sensitivity and specificity. But the applications of biosensors extend far beyond in the healthcare domain. In the environment, biosensors are employed to detect and quantify environmental pollutants, toxins, and contaminants, contributing to the monitoring and protection of the environment. In the food industry, biosensors are used to detect foodborne pathogens, impure foods, and spoilage indicators, ensuring the safety and quality of food products. And in forensics, these devices can be used to detect the presence of explosives, chemical warfare agents, and other illicit substances, aiding in law enforcement and national security efforts. Due to the wide range of applications, these sensors come in a variety of forms, each tailored to address unique needs and challenges. Currently, there are three primary types of biosensors utilized: enzymatic, antibody-based, and nucleic acid-based. Enzymatic biosensors use enzymes, which are biological catalysts that can recognize and interact with specific molecules. These sensors use enzymes' high specificity and sensitivity to detect the presence and concentration of the target analytes. When the target interacts with the enzyme, it triggers a measurable change, such as the production of an electric signal or a visible conformational change. This signal can then be amplified and translated to quantifiable data. Antibody-based biosensors, on the other hand, use antibodies as the recognition element, which can detect the presence of a wide range of analytes, from small molecules to large biomolecules. The binding interactions between the antibody and the target generates a measurable response, such as the change in electrical signal or mass-related properties. Nucleic acid biosensors employ oligonucleotides that can hybridize with complementary target sequences, enabling the detection of specific genetic markers or pathogens. Nucleic acid-based biosensors offer great specificity and sensitivity, making them extremely useful in disease diagnosis, genetic analysis, and environmental monitoring. In today's video, I wanted to highlight a unique type of nucleic acid biosensor known as aptamer-based biosensors, and how the Hong Kong CityU-HongKong team are leveraging this to fight a critical battle in pharmaceutics. Who are you and what does your project do?
We're very grateful that you can have us today. We are a team of students and researchers from the City University of Hong Kong, specializing in biomedical science and biochemical engineering. Our project focuses on developing a platform to combat the circulation of counterfeit bevacizumab on markets. This issue is developing, especially in sensitive regions. So specifically, we are working on the development of a biosensor device using aptamers that recognize bevacizumab, this anti-cancer drug, antibodies they are, via a lateral flow assay. So this will enable detection of the drug's concentration and purity. So our goal is to collaborate with drug authorities in the future, with pharmaceutical companies, to integrate our product along with the production line of the bevacizumab drug kits. So our device could be also employed by individuals such as healthcare practitioners or patients themselves to secure the drug authenticity, thereby ensuring the security and authenticity of bevacizumab against counterfeit drugs on the market.
That's awesome. So it seems like your idea is to kind of create a method for rapid detection of counterfeit anti-cancer drugs. So how did you formulate this idea and what kind of led you to this development?
So bevacizumab, as I mentioned before, is an anti-cancer medicine that inhibits angiogenesis. So angiogenesis is a common physiological mechanism in advanced stages of progressive cancers. Bevacizumab can thus be applied as a treatment to many common cancers such as colon cancer, lung cancer, renal carcinoma, ovarian cancer, etc. So we can sure see the treatment value in bevacizumab. However, this drug is not really universally approved everywhere, nor could it be accessible for every cancer patient since it is in advanced stage chemotherapeutics. So there was this oncologist that I talked before. She's from Armenia. So she addresses how in Armenia, bevacizumab is not really approved or covered in insurance or used in clinical practices nor provided by the government by any legitimate authorities. So patients they would need to go out their ways to acquire these drugs themselves, and particularly these methods would require them to get counterfeit drugs or from the black markets, and this is when and where counterfeit drugs step in and become the largest concern. So I think in the past, US FDA, they reported the finding of probably around 19 medical practices that bought and used counterfeit versions of Roche, the actual or the largest pharmaceutical company, their version of bevacizumab. And this comes into concern for the public and especially cancer patients and family members of whom, whether or not the drug that they required are worth the cost and are they up to the optimal quality for treatment. So what our group strives to achieve and discover is a method that is stable, rapid, and low expense, precise, sensitive, and high efficiency in targeting these bevacizumab contents in these drug kits. So our device is a aptamer-based lateral flow assay. So why we use aptamers is because aptamers are short single-stranded nucleic acids of DNA or RNA, or they could be peptides. So they are akin to antibodies in their binding capabilities and target specificity. Yet compared to conventional detection assays that are antibody-based, the aptamer-centric assays, they hold like better chemical stability and longer shelf life. They're portable, they're very easy to manipulate, to use, to apply, and also they're very much lower cost for production compared to antibodies. So conventional methods may require the use of antibodies, which would require a much more complex procedure for the detection or the experiment testing procedures. Whereas aptamer-based lateral flow assays, they can yield results within minutes and with higher precision.
Traditional nucleic acid-based biosensors rely on the use of nucleic acids as the recognition element. The primary difference lies in the nature of the recognition molecules and the selection process. Traditional nucleic acid-based biosensors typically employ naturally occurring DNA or RNA sequences that can hybridize with complementary target sequences and are often used for the detection of specific genetic markers. Instead, aptamer-based biosensors use synthetically made single-stranded DNA or RNA aptamers that are adapted to bind to a wide range of analytes, from small molecules to whole cells based on their physical conformation. Aptamers' ability to form these vast varieties of shapes with high selectivity and specificity is due to their tendency to form helices and single-stranded loops. Rather than primary sequence, aptamers' binding is determined by its tertiary structure. For aptamers to recognize their target and bind to analytes primarily depends on the three-dimensional shape-dependent interactions. Aptamers bind if they fit their target. Here is the Celix process as depicted by base pair technology. To begin, a single-stranded RNA or single-stranded DNA library is placed into an affinity column, with the analyte structure held in place. Oligonucleotides that bind will stay on the column, and those that don't will fall and be discarded. Using an elution buffer, the desired oligonucleotides are kept and amplified using PCR. This iterative process is then repeated with increasing stringency to enhance enrichment of the oligonucleotide pool using both positive and negative selection. Around 6 to 10 rounds of CityU are performed, yielding roughly 10 to the power of 6 aptamer sequences. Due to the adaptive nature of aptamers, aptamers can bind to small molecules, to proteins, to cells, and even organisms. These include aptamers targeting PSMA, prostate-specific membrane antigen for prostate cancer diagnosis and treatment, and cell-specific aptamers for targeted drug delivery, cancer therapy, and cell isolation or purification. Aptamers can even bind to detect specific minerals, ores, or rare earth metals. Let's hear from the CityU team what they are using aptamers to target and how it helps them identify counterfeit cancer drugs.
So we're talking a bit about aptamers and introducing aptamers to the audience. We also kind of defined the CityU process. So could you walk us through your CityU process and how you used it to identify aptamers for your project?
Sure. So CLEX is, well, for long, it's systemic evolution of ligands by exponential enrichment. It's a method for selecting aptamers with high affinity and specificity for target molecules. So the CLEX process involves generating a large library of random nucleic acid sequences, and then we incubate them with the target molecule. In our case, we would be incubating it with the bevacizumab drugs. And partitioning these mixture. So in this mixture, there would be bound sequences. This bound sequences would be the oligos bound to the bevacizumab, and then the unbound ones. And so partitioning process would then exclude the bound ones that has our target. And so we would then isolate the bound sequences, and those oligos would become our aptamers.
Yeah, thank you. So I think you talked a little about how Vakazuma is the specific analyte that you're targeting. But how does that play into like the general project goal of identifying counterfeit cancer drugs?
So bevacizumab is our target analyte that will be used in CLEX. And the products after CLEX enrichment are the oligos that we will be monitoring for optimal stability and concentration application and in vitro testing for possible modifications and such. So our major focus would be working on molecular chemical modifications of the aptamer to see whether we can manipulate the interaction or the binding affinity between the aptamer and the bevacizumab, the antibody that we will be working on. So with this mechanism at hand, at a settled level of concentration on the test strip, the aptamers are able to bind with high affinity to the bevacizumab drug, if any are present in the sample at test. Basically, the counterfeit drug that should be containing bevacizumab would be applied as the sample to run down the membrane strip of our lateral flow assay. And if any presence of bevacizumab would be detected if there is any at great sensitivity and signaled by this display of the test line. But if the counterfeit drugs are in fact lacking bevacizumab, controlled DNA probes that we will also be using would compete over bevacizumab to bind to the aptamer and show two lines suggesting negative presence of the bevacizumab drug, thereby indicating, oh, this is a counterfeit drug.
However, simply identifying aptamers that bind to bevacizumab is not enough to tell consumers which drugs are counterfeit. A test system that can utilize aptamer technology and allow consumers to interpret the results are necessary to create a functioning product. Here's CityU's solution.
Why specifically did your team choose to develop it using a lateral flow assay format for your aptamer-based biosensor?
So it's basically because like our main focus of like our team is not basically just like identifying like whether this drug is like fake or real drugs. But we also want to make sure that this drug is like accessible in like all places without needing like any sufficient technical expertise of like a specific stuff, or also like needing like any kind of like specific like medical tools. We want to create like some devices that can be easily distributed, which is like the main reason we choose like the lateral flow assay that can provide a quick, cost-effective, and user-friendly diagnostic tool that can be used by like anyone at any time.
So knowing this, how do lateral flow assays in general work? Lateral flow assays are a type of rapid diagnostic test that have become increasingly popular for point-of-care testing such as COVID-19 or pregnancy tests. These simple paper-based devices work by using capillary action to draw a sample such as blood or urine through a test strip containing specialized reagents. Unlike PCR tests, which require sending patients' sample to the laboratory and involve complex laboratory equipment and highly trained staff, lateral flow tests can be processed on the spot and done at home. Lateral flow testing kits are small, portable, and administrable by anyone with minimal training. This makes them attractive both for consumers who want a quick detection system and for countries rolling out mass population screening programs. Let's hear how the CityU team's aptamer-based lateral flow test works and how they plan on distributing this technology.
But to simply like speaking, like this design that we're making is we will like apply the aptamer by immobilizing it on top of like the test strip pad. And in application, the sample will be applied to the test strip sample pad and then migrate through the pad and encounter a gold nanoparticle conjugated with a form of bevacizumab. If the bevacizumab is present in the sample, it competes with the conjugated form for binding site on the membrane, and this competition prevent the gold nanoparticles from binding on the test line, resulting in a lighter or absent signal at the testing line. The test strip itself also include like a control light that captures the gold nanoparticles to ensure the test functions correctly. The result will be shown in like two ways. The first one will be like one line that only shows the control line, indicating that the bevacizumab is present in the sample. But the test line is not visible because the analyte from the sample effectively competes with the labeled form, preventing it from binding to the test line. While the other result will be like two lines that shows like both the control line and the test line, indicating that bevacizumab is absent or present in insufficient amount to compete. And both lines are visible because gold nanoparticles can freely bind to the test line.
How do you see this technology being implemented, like on a wider scale? How do you see maybe your distribution of your product?
Yeah, we do have like a broad view of like how this technology can be implemented in the futures, but for like in general, like the aptamer-based LFA can be applied in various fields, including like healthcare and pharmaceuticals to ensure the quality and the authenticity of medications, such as for like quality control. It can also be used in like clinical settings, such as pharmacies and the point of sales to quickly verify the legitimacy of the drugs. Uh, by like doing like this, we hope that it can prevent the distribution of the use of like the counterfeit products to the patients. And additionally, this technology can also be applied to adapt for like other kind of like biomolecules in the future. So we hope that other research group maybe can also implement this for like other types of cancer drugs, since our focus now is only for bevacizumab. But in our specific case, we aim to distribute this test strip to various regions, particularly those vulnerable to drug counterfeiting, such as conflict zones and developing countries. And by partnering with like various like health organizations and also physicians, we can ensure that the proper use and distribution of this test strip will improve the quality of cancer treatment in the high-risk area.
As aptamer research and development continues to advance, we are seeing several exciting emerging trends that point to the future direction of this technology. One key area of progress is in aptamer engineering and the Celix process used to identify and select aptamers. Researchers are exploring new Celix strategies, such as cell Celix and in silico Celix, to expand the repertoire of available aptamers and target even more diverse and complex analytes. Additionally, advances in computational modeling and machine learning are enabling more efficient aptamer design and optimization without the need for continuous iterations. Researchers are also beginning to develop ways to use multiple aptamers simultaneously. By combining multiple aptamers that can bind to different targets, researchers are creating aptamer panels that allow for simultaneous, high-efficiency detection of various analytes. Another area is how aptamer research is being adapted to new technologies. The integration of aptamer technology with emerging platforms like microfluidics and smartphone-based devices is also gaining momentum. Microfluidic chip-based aptamer assays enable small, automated aptamer tests, while smartphone-integrated aptamer sensors leverage the prominence of mobile devices to bring diagnostic and monitoring capacities directly to the user. Looking ahead, the versatility and customizability of aptamers is bound to have a significant impact in various sectors. In healthcare, aptamer-based diagnostics continue to advance, with potential applications in personalized medicine, drug delivery, and therapeutics. In the environmental and agricultural domains, aptamer-enabled sensors can contribute to improved monitoring, risk assessment, and sustainable resource management.
Great, thank you so much for sharing. So in terms of the next question, so you spoke a bit about how the technology can be applied to detect other biomolecules and make it more versatile for diagnostic applications. So how do you see the future of aptamer-based biosensing going? Like, where do you see this direction of this emerging field going in maybe, let's say, the next 5-10 years?
So, in my opinion, like in the next 5 until 10 years, we estimate that the aptamer-based biosensor will likely become a fast solution for tackling various global issues and advancing synthetic biology. Since like for now, we are focusing on like targeting like discovering whether a drug is like a counterfeit or not. But actually, there's like various other application that has like utilized like the aptamers, such as like for environmental costs, like targeting like whether there's like a pollutant in water and like a lot of like other kind of like cases that people want to see and use like this aptamer for like a more effective results. But we can also see that recently there has been like a lot of like improvement in terms of like the aptamer development by using like the artificial intelligence. AI will make this biosensor more reliable and also speed up like the development process by optimizing like the aptamer selection, for instance. As this technology evolve, we should see like easier customizations and also better performance for various application in the near future.
In conclusion, aptamer-based biosensors represent a transformative technology with the potential to revolutionize a wide range of industries and applications. The unique properties of aptamers, their ability to bind with a diverse array of targets with high specificity and affinity, in addition to their chemical stability and scalable production, make them extremely versatile in recognition element of biosensor development. With aptamer-based biosensors heading in an upward trajectory, let's ask our team some final questions and how they see their own project developing in the future.
So what are the next steps for your project in consideration? How do you see your own project developing in the future as well?
So currently we are like focusing on like trying to make like the proof of concept of our project, but in the long-term goal, we would like to get like the beta test strip to have like a regulatory like approval for testing in diagnostic centers, labs, and also hospitals. And in the future, we plan to send this test strip to region most vulnerable to counterfeiting, including like the conflict zones and the developing countries that I might also like mention like previously. We aim to work closely with like many more like NGO and like physicians to ensure that the distribution and proper use of this test strip can really like benefit the patient directly and make sure that we will have like a decreasing like trend in terms of like counterfeiting drugs in for like cancer patient like treatment in like various countries worldwide.
That's awesome. So since this is a video targeted towards kind of high schoolers and undergraduates who might be interested in emerging fields in synthetic biology, what key takeaways do you think you want the audience to have to remember about your team's project? Or
Yeah, that's a very good point. So throughout like all of like this talk, we have been like talking about like counterfeit drug. We talk about like the biosensors, and we know that counterfeit drug, especially like fake anti-cancer drugs such as bevacizumab, can pose serious health risks by compromising like treatment efficacy and making therapies less effective. Identifying this kind of issues really want us like to create something and get like a very like valid solution, such as using like the biosensors that can address the issues by offering a quick, cost-effective, and user-friendly method to verify like the drug authenticity. This innovation that we're creating will aim to like enhance patient safety and also improve treatment outcomes and significantly impacting healthcare by ensuring patient receiving like a genuine and effective medication and getting something that is on par with what they are already like spending for like the cost of medication. So like if I need like to really just like summarize everything in one sentence that you should be able to remember is that the technology that we are creating will we will ensure it so that it can like spot the fake and also like safeguard the cure, which is also like our main goal in like throughout like all of this like iGEM project.
Yeah, so that's awesome. I love your team motto. So how can others like kind of get involved in this field or learn more about developments? If you want to share, you can also share maybe your team Instagram so people can keep in touch or ask you questions maybe. But yeah, like how do you think others can get involved?
Yeah, sure. So like for those that would like want to know more about like what our team has been doing and what kind of like our improvements, such as of like the expert interview result that we did like with various experts and also the result of our project, you can just like we will provide like the barcode and you could just like scan like the QR code to follow like our Instagram, also like our LinkedIn page. But for all of like the high schooler or like the undergraduate students that is like interested in this field and want to learn more, I will encourage you by starting to explore the current like issues. Like it's very important to know like what kind of issues you are trying to solve and how crucial these issues is to our like global community. And then you could also explore more about like the current biosensors like trend, what kind of technology other people have been like developing and how like this technology can help to solve like each issues that you already identified before. So it's really important for you to stay updated with the technological advancement and engage in hands-on research like wet lab work or like simulations when you have like the chance in your school maybe. And it's also like another like very important like point when you can get like the chance to connect with communities like iGEM community that have like a lot of like-minded like researchers that can help you to improve your research project and also like open your scope of like research to like a wider scope in the futures. And lastly, like don't hesitate to seek advice for like from like experienced professionals like your teacher, mentors, professors, or any other people that will definitely like likely to help with your project, because like we really believe that throughout like all of this competition, like what we are doing right now through like this collaboration, like collaboration is indeed like something that is very important in terms of like innovating something new and solving like a very crucial like global problem like the drug counterfeiting around the world.